Well, thank you very much for the invitation to give this talk. I'm Alvaro Orsi. I'm a principal scientist at PlantTech. And just a bit of my background. So before joining PlantTech, I was actually doing computational cosmology before coming here. So, some people say that Earth observation, remote sensing is like astrophysics, but turned upside down, essentially. So I think there's a bit of truth in that. And what I'll be showing you here is really the result of collaborative effort between, by a very large group, as you can see. So we've got Ian and Reddy. Ian has already spoken, Reddy will be speaking later, Mohammed, Ishvan, and Andrew Gilman from PlantTech, and then people from Plant and Food Research, from Eurofins, Durham University in the UK, and the University of Melbourne in Australia. So I'll be just mentioning three things. So first, the motivation of this work that combines LiDAR and hyperspectral imaging. Then I'll be just discussing a little bit about these two platforms in the context, and then I'll be talking about 3D relative transfer modeling, and that's sort of my talk. So let me just first motivate this idea. What I want us to think about now is essentially what is really going on when we discuss something like the spectral reflectance, right? For example, in this case, of a canopy, of a vegetative canopy, right? So essentially what's happening is that the vegetation canopy is receiving solar radiation, and it is in some way responding to it, right? If you think of it at the photon level, then there are mostly three different possibilities. Photons might be reflecting off a surface, in this case leaves. They might be transmitting through the leaves, or they might be absorbed, right? When we discuss leaf and different leaf properties, we can use different relative transfer models that try to encapsulate the relation between biophysical and biochemical parameters and these three optical properties of leaves. Interestingly, these vary significantly with wavelength for leaves, and as I was saying, depend on biophysical and biochemical properties. Okay. Now, there's really a fourth channel, if you like, which is what happens when some of the radiation is absorbed, but then some of it is essentially dissipated or sent back into the environment, right? And that is essentially our goal in this project. So if you look at this diagram here on the bottom left, it's showing how from some incident light that is interacting with a leaf, some of it will be absorbed, and this is essentially radiation in the PIR region, so it's roughly from 400 to 700 nanometers. But then some of that radiation, well, some of the excess radiation will be reflected back, some of it will go through the leaf as well, and most of this radiation will contribute to the photochemistry, to photosynthesis, right? But essentially not all of it. So the light receptors become saturated very quickly in the leaf. And so the leaf needs to dissipate some of the excess energy back into the environment. And there are two main mechanisms. One is heat dissipation. That's sort of the preferred way for leaves to dissipate energy. But the other one is chlorophyll itself has an excitation process that emits fluorescence. Okay, and interestingly, when you look at chlorophyll fluorescence, you get a really good. Chlorophyll fluorescence can be thought of as a really good proxy for understanding the photosynthesis process. And the good thing of this is that now you can essentially use chlorophyll fluorescence, if you're able to measure it, as a benchmark for photosynthetic efficiency, sorry. So here on the right, I have an example of some grapevine leaves. The one on the left is a control leaf, and the one on the right is infected by a particular disease. And you see these are different fluorescence and photosynthetic properties that you can derive from chlorophyll fluorescence. And you see how both vary significantly. And this is because essentially any source of stress in a plant will affect the photosynthetic efficiency of it. And so chlorophyll fluorescence becomes a really good early signature of plant stress. And that is essentially our goal, right? Now, this is really, one thing is to measure this in a lab, in a controlled environment. But when you think of it from the remote sensing perspective, this becomes an incredibly difficult measurement to make. And the reason is because chlorophyll fluorescence, or sun-induced fluorescence in this case, is an extremely weak signal. It accounts for just a few % of the radiation in these two narrow wavelength ranges, right, as you can see in the image there. And the other thing is that if you now want to use it as a signature for plant stress, then you need to understand what that fluorescence is really telling you from a photosynthesis perspective. And that requires understanding the radiation transfer process and the photosynthesis processes in the canopy as well. So our strategy to do this involves an extensive data collection campaign, which consists on acquiring airborne LiDAR scanning data with a drone, also an airborne hyperspectral imaging sensor like the one Ian described earlier in his talk, and also an extensive ground truth field collecting experiment. We want to put everything together into a 3-dimensional radiative transfer model. I'll explain in a second why. And all of this is, the whole processing of this is very computationally intensive. And so we need to use deep learning models that will essentially optimize our parameter search so that we can constrain what happens. How do we interpret fluorescence in terms of photosynthetic efficiency and thus plant stress, okay? So now let me go to the remote sensing platforms. The first one I want to describe is LiDAR. So we want to do this, well, we are doing this from with a UAV instead of ground truth LiDAR sampling. And the reason is essentially that we want to cover a lot of area. But doing this from above, from the top of the canopy, has got many challenges. Includes many other challenges. So that is an example of a point cloud of a kiwifruit orchard. And what we really want to understand now is how can we extract from this point cloud all the canopy structural parameters that will be key to understanding how radiation is interacting with the canopy, okay? So what we are doing to do this is our goal is to reconstruct the canopy structure from the LiDAR's point cloud. And what we're doing here is that instead of taking trying to measure some of these parameters directly with ground truth sampling, we are actually trying to simulate how a virtual canopy would look like, and how measuring, creating a point cloud with a LiDAR sensor in a simulation would capture that, you know. So we are using procedural tree generation models that resemble a kiwifruit tree, and that's what is shown there on the left. And we are using a LiDAR simulation plugin on Blender. So Blender is a 3D modeling software. And as you can see in this animation, we've got the tree, and then we simulate how an aerial sensor would be collecting point cloud data. Here on the left, in the bottom, I'm showing the resulting point cloud. And then the idea is to learn from this point cloud, how can we extract things like the leaf inclination area distribution, and this is what it's shown here in the middle, or the leaf area density, which is sort of a differential version of the leaf area index. Okay, so we can do these things applying different techniques that utilize not only the point cloud position, but also the relative ray path, essentially, between the sensor, or from the sensor to the location of the point itself. And these plots are comparing our infer values for the leaf inclination angle and the leaf area density against what we can read directly from the simulation, because the simulation we can control these things to make them look whichever way we want. So in this way, we can create algorithms that are very that can adapt easily to essentially an infinite variety of conditions, and that's what we're doing. Then the other one is to use the airborne hyperspectral imaging data to extract sun-induced fluorescence. So this is using the same sensor that Ian was describing before, and that's an example of the reflectance data that we are obtaining, right? And maybe I should go back for a second. Essentially, the signature for sun-induced fluorescence would be somewhere very near the red edge that you see there, close to 700 nanometers. So actually, if you look at here on the bottom plot, the red curve is an example of how fluorescence looks like. And as you see, it goes roughly from 600 to 800 nanometers, but it's got two peaks, which are really the only thing that you can typically measure. It's got one peak at 680 more or less, and another one at 740. And we use Fraunhofer lines that come from the solar spectrum to essentially see what is the impact of fluorescence by diminishing the trough created by these Fraunhofer line detections. Right. So we can also simulate this and look at the impact of the spectral resolution of our instrument and then we essentially try to implement different algorithms to extract fluorescence from these Fraunhofer lines, and we can see, well, in this plot, essentially we can see that our sensor, which has a full width half max of 3.2 nanometers in this range, can actually recover the fluorescence peaks reasonably well. So that's part of the work. Now, why are we doing this in the context of 3D relative transfer modeling? And this is because sun-induced fluorescence is such a weak signal. We want to make sure that we are modeling the emission of fluorescence in the canopy in a very robust and accurate way. And for that, we need to make sure that we are able to incorporate all the different scatterings and transmission through a very complex canopy. So that's why we use the structural parameters that we read from the LiDAR service are then used to generate a virtual canopy. And this is what it's shown here. And this is input in a 3D modeling framework called Helios. And then from here, we can now simulate the interaction of the canopy with sunlight, but now using ray tracing. So ray tracing is the same essentially simulation, if you like, or the same technology that is used in modern video games, right? Which literally simulate the paths of individual rays or photons as they interact, with your 3D scene. Okay, but now we can incorporate how biophysical and biochemical parameters have an impact on the reflectivity and absorptivity of leaves. We can actually do this at the individual leaf level, and this is showing an example, right? So you see
2 minutes.
Yeah, thanks. And so you see that now our simulation will depend on all those parameters that can be measured or can be inferred. And the fluorescence, for fluorescence, we can use yet another fluorescence relative transfer model, which now depends on yet another set of parameters that we can measure or we can infer as well. And so in the end, with this, we can now predict what the sun-induced fluorescence will be and how this compares essentially to our to the actual measurements that we can extract from the airborne hyperspectral sensor. The end result of that is that by exploring the parameter space that results from all these biophysical and biochemical parameters that are unconstrained, if we can do this in an optimal way, we will get the best parameter estimates, which result in essentially stress maps. And what's more important is that we will also learn what are the insights or what are the drivers of stress, right? And this is essentially the main goal, because now we can discuss not just we can say not only that there are regions in the orchard that might be suffering stress, but we will also be able to tell the growers or the orchard managers why there is stress in that particular region. Is it a lack of water? Is it a lack of a particular nutrient, and so on? Okay, so those are my conclusions, but I'll just stop here. Okay, thank you very much for listening.